I'm going to leave this area, but we'll come back when we talk about the specifics of the coronavirus exocytosis pathway. So for now, if you can just imagine an RNA molecule coming along here and essentially just inserting itself into this region of the ergic and grabbing this membrane to engulf itself and picking up spike proteins and some other membrane proteins on the way. That's pretty much what happens, and then the rest is going to be stuff that we'll talk about eventually later on. But for now, let's get back to the business and wrap up the business of secreting our collagen or whatever it is that we wanted to send through the secretory pathway, talking a little bit about the processes that go on here and just slightly beyond. So to do that, here's a nice diagram from your text that is an artist's reconstruction of the tubular network that is the Golgi apparatus from cis to medial to trans. And here is an actual electron micrograph of the same thing. So if you focus for a second on this secretory vesicle right here, then in terms of the mechanistic aspects of that process, that would be putting us down here. So we've managed to get our soluble luminal protein here using the trafficking and sorting pathways up to this point. And then there is a series of reactions and maturation processes in terms of the soluble cargo itself and also the compartments that it's in that takes us from the cis through the medial to the trans Golgi. If you've read the textbook on this section, then you'll realize that this mechanism, as far as the organelle is concerned, is very controversial, and no one seems to agree on the exact process by which it happens. We're just going to say that a miracle occurs and our cargo is now here, and then what happens next, and we won't talk much about that either. But the possibilities are that it is inserted into one of those secretory vesicles and then will eventually fuse with the plasma membrane and released as soluble extracellular membrane components like collagen, elastin, stuff like that. It's also possible that, as I mentioned, it could be the GLUT2s of the world in which it is targeted to the plasma membrane where it functions. And then there is a sorting pathway that we're not really going to talk about that would put it in the lysosome. There are pathways that move in this direction that we'll talk about in the context of endocytosis. About endocytosis, it is a huge topic, and I'm going to chunk it a little bit. I'm going to, in this video, just treat it like a standard trafficking reaction, really talking about the budding process that occurs at the plasma membrane to bring in the first vesicles. You can probably get an idea from this diagram. There's a lot going on, and much of that fits under the general category of endocytosis, and I'll talk about those reactions in the next video. By the time we talk about those, we're really talking about things that go beyond simple cargo movement and involve much bigger processes. But for now, just to keep things a little bit simpler, I'm going to treat it like a standard vesicular trafficking reaction, in which case we talk about coats and adaptor proteins and stuff like that. So in that vein, as I've mentioned earlier, this region from the plasma membrane to the trans-Golgi network is generally speaking the domain of clathrin as the coat. So this is what clathrin is. It's a large multimeric structure built of 3 heavy chains and 3 light chains that if you take a bunch of these and put them together, you create sort of like a puzzle that you would get at a gift shop. And that puzzle ends up being a sphere that's almost identical in structure to a soccer ball that uses hexagonal and pentagonal panels that just alternate. That's exactly what the clathrin coat looks like. It's made up of individual structures, not monomers, but individual pieces. And here's probably a cryo-electron micrograph of that structure, and they're referred to as triskelions. So that's the sort of the repeating unit of the clathrin coat. And the reason it's called a triskelion is because there's an ancient form of art that was very common during one period of human existence, in which there was a face with 3 legs attached to it that would appear over and over again in the iconography of the cultures, and that was referred to as a triskelion. So it's actually an art term. But because this structure resembles that, at least superficially, the term was applied to the Clathrin repeating unit. So the way that works in terms of the budding reaction is shown down below. So we begin with cargo and receptor. So for some reason they don't want to admit to the fact that there are cargos that are integral membrane proteins, but same rules apply in endocytosis. So here is the cargo receptor bound to an adapter protein. So this is going to be a trafficking motif here that is recognized by this thing. And this is a simplified version of what happens, but the idea is correct. The cargo molecules and receptors, if there is one, and adapters form a cluster at the surface of the plasma membrane. This could also be in the trans-Golgi, which is why it's kept somewhat generic. We're calling it a donor. But let's just talk about bringing something in from the plasma membrane. That will then recruit the triskelions. They accumulate by attachment to the adapter proteins, which of course are adapted to the cargo either directly or indirectly, and that leads to the formation of a Clathrin-coated pit that pinches off near the surface of the plasma membrane and becomes a Clathrin-coated vesicle, and this is the soccer ball. That is the Clathrin triskelions arranged in those patterns. The sandwich contains the adapter proteins and all the other stuff that we talked about earlier, and then some. This is a very, very complicated trafficking vesicle compared to COB1 and COB2, but the general idea is the same. controlled uncoating of the now naked transport vesicle that then goes on to do either pretty standard translocation infusion if it's going to the trans-Golgi, or maybe if it's going to the lysosome. That's not too unusual, but there are a lot of other things that can happen with this transport vesicle that makes endocytosis a much more complicated topic, and again, we'll cover those in the next video. But for now, I just want to kind of wrap up the cargo business in terms of vesicular trafficking with this diagram that introduces something that I haven't talked about, but really is a theme that operates all through vesicular trafficking. This is just the trans-Golgi to plasma membrane to perhaps the lysosome directions. But what's shown here is that trans-Golgi and the trafficking vesicles that come from it. So here's something that's related to the last diagram with the cartoon of the Golgi. Two forms of exocytosis. This one goes on continuously, and this is how something like collagen would be secreted into the extracellular matrix. Regulated exocytosis, this cargo would probably be something like a neurotransmitter that goes to the surface and sits there. It actually goes through the tethering and docking stages of that three-step fusion reaction and then stops and waits for a signal that then allows that final fusion reaction to occur, and then neurotransmitters are released. Talk more about that when we talk about nerves. Then going back to endocytosis, here is a reaction that produces a trafficking vesicle that becomes probably a late endosome at this stage, and then something that we'll talk about in the next video, either an MVB or a lysosome, and ultimate destruction. And it sort of ties into phagocytosis, which we're not going to talk about at all at the level of the lysosome. But the real point of this diagram is these color-coded things. They are the different forms of phosphatidylate and non-acetyl phosphate that are specifically associated with vesicular trafficking. That is, there are specific regions of specific membranes or vesicles that play regulatory roles in dictating the overall direction and the coherence of the vesicular trafficking. So that, for example, the types of vesicles that are produced for regulated versus constitutive exocytosis would differ in terms of the phosphatidylinositol concentration. And the process of endocytosis that's leading to this thing that we just talked about would be associated with this particular type of phosphatidylinositol. And this is phosphatidylinositol 4,5 diphosphate, so PIP2, but PI45P2, which is going to be distinct from another P2 that is got phosphates on different positions of the inositol ring. So you may have heard about PI3 and PIP and IP3 in other classes. So this is all related, but these are different intermediates. This is a PI3, this is a PI3, and that would be a PI3 kinase associated concepts that we'll get to eventually. But the idea is that these different forms of phosphatidylinositol serve as signals to dictate which type of vesicular trafficking occurs and where it occurs. And this diagram is meant to indicate that there are patches of PI45P2 that are found dispersed throughout the Golgi apparatus, and that is part of the regulatory processes that dictate where there's going to be a clathrin-associated trafficking event. How that works is known pretty well in the case of endocytosis, and not so well with the other trafficking reactions, especially COP2. It's known that PI is involved, but exactly how is not well understood. But in the case of endocytosis, the way it works is pretty clever. So you have an adapter protein here with binding sites that eventually are going to recognize the trafficking motifs of the cargo receptors. So this would be the NPXY or the YXXV of a table from a couple of videos ago. So at this stage, the adapter AP2 can't bind, but if it associates with the appropriate phosphatidylinositol, and that means that this has to be clustered before anything else happens in order to say, begin endocytosis here. If that happens, then you get a conformational change that opens up the binding sites of AP2 so that it now, in complex with the phosphatidylinositol, can make those contacts with the cargo or cargo receptors, and that brings clathrin in, and the rest is as is shown on that previous diagram.